Thump, 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 yeah. So you know what to do with this thing? Throw it away or turn it off. Well, good evening. It's a pleasure to introduce our visiting speaker, Dr. John Shao. He will be speaking about the role of abnormal proteins in the Alzheimer's disease. We have a good crowd tonight. I think you'll really enjoy this talk, and it'll be educational. Dr. Shao is the director of the Diagnosis and Biomarkers Program, which is in the Neuroscience Division of National Institutes of Aging. He holds an MD from the University of Maryland. He's been trained in psychiatry at the University of North Carolina and also at the National Institutes of Mental Health, where he led clinical trials of mental diseases, including schizophrenia and Alzheimer's disease. As a fellow at NIH, he completed training in nuclear medicine, which is fundamental in interpreting the specialized brain scans which you'll hear about tonight. Dr. Shao is a board-certified psychiatrist. That had to be handy. He also served in the public health services for 30 years and has authored or co-authored over 90 scientific papers. Our speaker tonight was brought to us by Diamond resident Jim Bailey. Unfortunately, Jim was not able to be here tonight. Please welcome our speaker, Dr. John Shao.
Okay, and you can hear me? All right, thank you very much. It's a real honor to be here. You'll notice that the title of my talk is a bit different than the syllabus. It's not just focused on diagnosis, although I will mention that briefly at the end. This will really be about recent progress in Alzheimer's and some good news for a change. We need some good news. So I don't have really any disclosures to make. I do want to point out that these views are those of the speaker and don't necessarily represent those of the NIH or the United States government. No real disclosures. I have a limited solo private practice. It's getting smaller and smaller. But there's been a lot of progress in Alzheimer's that the public isn't aware of, and I believe that we may actually be on the brink of a major breakthrough. What you need to know, though, to understand that is some of the background, how we got here, some of the history and biology of Alzheimer's dementia, and in particular, how we developed the amyloid hypothesis, which I think most of you have heard about, but has become controversial lately. It isn't wrong, but it's incomplete. This is important because we finally have, some disease-modifying treatments based on the amyloid model that I'll be talking about. However, preclinical Alzheimer's shows that amyloid isn't the direct cause of dementia. There's another protein, tau, that most of you haven't heard about that directly causes dementia. Alzheimer's has two phases, a pre-symptomatic and a symptomatic phase. And an important question is whether treatment during the pre-symptomatic phase could prevent dementia, and finally I will talk briefly about a blood diagnostic. So an important reminder, Alzheimer's is not the same as dementia. Alzheimer's is a dementia, but it is a specific type of dementia. Many people use the terms interchangeably, but Alzheimer's refers to a specific dementia that is related to plaques and tangles, which I'll be talking about more. Dementia, just to remind you, is unexpected loss of cognitive ability associated with aging and neurodegeneration, brain shrinkage. Alzheimer's is the most common dementia, but it isn't the only aging-related dementia. There are others, Lewy body dementia, vascular dementia, TDP-43, and these become more important as we age, and particularly as we get in our 80s and 90s. What you can see in this figure here is that at older ages, people have multiple neuropathologies that it's very rare to have just Alzheimer's plaques and tangles neuropathology as people get older, or when people get older. So Alois Alzheimer was a German psychiatrist and neuropathologist. He ran an insane asylum in Frankfurt. In 1906, he presented the case of Frau Auguste D., who fell ill in her 40s Alzheimer's saw her when she was 51 and hospitalized in his hospital. She couldn't pay, so she agreed to allow him to examine her brain after she died. He looked at her brain under a microscope using the recently developed silver strains that Camilo Golgi had just developed. He didn't have photos, of course, but he drew pictures and he described. But in 1999, we were able to actually find the original slides that Alzheimer's had, and that's what's shown here in these two slides. And what he saw on the top there are the miliary foci, the round blob-like things that that he described as deposits of a peculiar substance in the cerebral cortex. And also in the lower figure, fibril bundles that were present as well. These are the plaques and tangles, and they are the hallmarks of Alzheimer's disease. And unless you have both plaques and tangles present, you don't have Alzheimer's disease. When Alzheimer's first named the disease, it really referred only to presenile dementia. And there is a form of familial inherited autosomal dominant familial dementia that begins in people's 30s and 40s. They develop plaques and tangles and dementia at a very young age, die by age 65. Frau D probably had this, and these are the plaques and tangles that Alzheimer's described. However, there's also senile dementia, which has the same plaques and tangles, the same neuropathology, and really the same clinical course, the only difference being the age. And senile dementia, of course, is in people's 70s, 80s, and 90s. There was a lot of discussion for a while whether senile dementia was more common or vascular dementia was a more common cause of dementia. But it really is senile dementia, Alzheimer's dementia, that is the most common cause of dementia. So in 1976, Dr. Katzman wrote an editorial in JAMA that described and argued that senile dementia is Alzheimer's. And he said Alzheimer's disease and senile dementia are a single process and should therefore be considered a single disease. Both Alzheimer's disease and senile dementia are progressive dementias with similar changes in mental and neurological status that are indistinguishable. The pathological findings are identical: atrophy of the brain, marked loss of neurons, neurofibrillary tangles, and neuritic senile plaques. The senile form of dementia may rank as the fourth or fifth most common cause of death in the United States. There were many hypotheses put forth about what causes dementia. One of the earliest was the cholinergic hypothesis. What was noticed in the 70s was that there's a marked decrease in neurons that use acetylcholine as their neurotransmitter in people with Alzheimer's disease. There's also theories about aluminum. There were reports of increased aluminum in the brains of Alzheimer's patients, but the epidemiology of this has really been pretty inconsistent, and it turns out aluminum is the third most common element in the world, and so it seems unlikely to actually be a major cause of dementia. Another theory is that Alzheimer's is caused by excess excitotoxicity or insufficient inhibitory neuronal activity, that the neurons are overactive. And this is still considered as a possible cause and still being studied. Finally, there have been a number of different infectious diseases that have been suggested as causes and possibly triggers of Alzheimer's, including herpes in particular, prions, and even dental plaque. So the point of any of these theories is to drive therapies. And the first therapies that were developed were the cholinergic inhibitors. Cholinesterase is the enzyme that breaks down acetylcholine, and rivastigmine, Exelon, donepezil, Aricept were developed in 1997 and 1998, and they block cholinesterase and increase the availability of acetylcholine, partially reversing the loss in cholinergic neurons. Memantine is another drug, Namenda, that is a blocker of the NMDA receptor and blocks excitotoxicity. Both the cholinesterase inhibitors and memantine improve cognition, but this effect is very transient. The time scale on these graphs here are weeks, not months. And the final result, which happens after many years, is the same whether or not cholinesterase inhibitors or memantine are used. What we really wanted was a disease-modifying therapy, a therapy that would affect the underlying biology that causes Alzheimer's. If we're going to do that, a good place to start is with the plaques and tangles that Alzheimer's described. Fortunately, there have been advances in silver stains that show the plaques and tangles better, and also the development of immunohistochemistry, antibodies to amyloid plaques and to tau tangles that can actually label the specific neuropathologies that are present. Both beta amyloid and tau plaques and tangles are amyloids. Amyloids are a class of disease that are aggregates of proteins that are stained by particular dyes. Pathogenic amyloids form when previously normal, healthy proteins lose their normal structure and misfold so they can stack up and aggregate into beta sheet secondary structure that basically stack on top of each other. You can see that in the figure here, and form these fibrils that then aggregate and form the plaques and the tangles that are found in Alzheimer's. On the right here is probably the most familiar amyloid, the prions. And prions are a cause of Creutzfeldt-Jakob, mad cow disease, and they are an infectious misfolded protein. And what happens actually is that the infectious prion causes normal prion protein, which is, we don't know what it does, but it's an important part of the body, to misfold and actually continue to aggregate and form these prion bodies that cause the death of the brain. Amyloid and tau, beta amyloid and tau aren't infectious, but they share many similarities with prions. One of the problems in studying Alzheimer's is that there are two proteins. There's beta amyloid and there's tau, so the scientists end up studying both of them. And when scientists study something, you know, they think that whatever they're studying is the most important thing in the world. And this has led to a long-standing tongue-in-cheek narrative about a religious war between Baptists studying beta amyloid versus Taoists that study the tau protein. The analogy that I like is to sports teams. You know, it's just a game, but the game is emotionally charged. Who's ahead, beta amyloid or tau? In 1984, Glenner and Wong were able to isolate the major component of the plaques that then found the beta amyloid peptide, which they called the A4 protein. In 1985, Inga and Khalid Iqbal showed that the neurofibrillary tangles are paired helical filaments made of the tau protein. In 1987, Colin Masters showed that the A4 protein, the beta amyloid protein, actually comes from another amyloid precursor protein that is a cell surface receptor. We still actually don't know what APP does, but that's where beta amyloid comes from. Tau, on the other hand, we do know very well. It has a well-known physiological function. It helps to hold tubulin together to form microtubules that provide the structure of cells and also provide tracks down which cellular cargo can be transported. Genetics can be very important to understanding disease. And causality can be hard to determine, but if you have an inherited disease, this can be made easier. If you look at the pedigree of someone, of a family that has an inherited disease, and if you compare patients that are affected with the disease to patients that are unaffected, eventually you can track down a gene that must be involved with the disease that only appears in the affected family members and not in the unaffected family members. Familial Alzheimer's is, as I said, an early onset form, beginning in their 30s or 40s, and children of people that have one of the familial Alzheimer's have a 50-50 chance of inheriting the disease. And if they inherit the bad gene, they will develop Alzheimer's in their 30s or 40s. If they don't, they will lead normal lives. Another condition that has associated with inherited Alzheimer's is actually Down syndrome. There's a very high incidence of Down syndrome, of Alzheimer's disease, plaques and tangles in Down syndrome. And this is a picture of somebody with Down syndrome, and you can see the characteristic plaques and tangles. Down syndrome is trisomy 21. There are 3 copies of chromosome 21. You'll remember that we have 23 chromosomes, one from our mother, one from our father, 23 pairs of chromosomes, one from our mother, one from our father. But in Down syndrome, there are 3 copies of chromosome 21, and it's believed that there's an extra copy of a gene that leads to Alzheimer's disease that's on chromosome 21. It also turns out that the gene for APP, amyloid precursor protein, is also on chromosome 21. In 1987, we showed that some families with familial Alzheimer's have a gene on chromosome 21, also have a gene on chromosome 21 that is causing their Alzheimer's. Now, all 3, you would think that this must be the same gene, but they really have to prove that, and that actually proved very difficult to do. And it took until 1991 to prove that the chromosomal 21 Alzheimer's gene was the same in familial Alzheimer's and in Down syndrome, and in fact is the gene for Alzheimer's precursor protein. That discovery prompted the proposal by John Hardy and Gerald Higgins of the amyloid cascade hypothesis, that deposition of amyloid beta proteins, the main component of the plaques, is the causative agent of Alzheimer's pathology. The neurofibrillary tangles, cell loss, and dementia follow as a direct result of this deposition. The rest of the 90s was spent on figuring out how beta amyloid gets cut out of APP. APP is an integral membrane protein that's concentrated in the synapses of neurons. It functions as a cell surface receptor, meaning that it signals from outside the cell to the inside of the cell, which is pretty important. It is cleaved by various different secretase enzymes, and it can either go into an amyloidogenic or a non-amyloidogenic pathway. If the first cleavage is by alpha secretase, it goes into a non-amyloidogenic pathway on the left there. If the first cleavage is by beta secretase or beta amyloid-associated cleaving enzyme, and then is cleaved by gamma secretase, beta amyloid is produced. You can see on the figure here that beta amyloid is sort of in the mid part of APP. We also learned that there are other causes of familial Alzheimer's that were due to other genes that were found on chromosome 1 and chromosome 2. These were named presenilins 1 and presenilin 2, and they were later shown to be both involved in gamma secretase and involved in actually cleaving the beta amyloid protein from APP. So this means that all the familial Alzheimer's mutations affect A beta metabolism. APP, the parent protein, which is also the cause of Alzheimer's and Down syndrome, or presenilin 1 or presenilin 2 that are part of the gamma secretase complex that create beta amyloid from APP. What you can see here is that there are multiple different mutations of each gene that have been found in families with familial Alzheimer's. Familial Alzheimer's is extremely rare. And NIH actually has a worldwide dominantly inherited Alzheimer's network, a multi-site study that tries to study these patients, tries to recruit them and follow them and learn what is causing their Alzheimer's. There's also mutations in the MAPT tau gene. However, these cause familial frontotemporal dementia, not Alzheimer's dementia. And this is, additional proof that really the central cause of Alzheimer's is beta amyloid, not tau. We also have a study called ALL-FTD that studies familial FTD as well as sporadic FTD. So the bottom line is beta amyloid causes Alzheimer's disease. That's that question. All mutations causing familial Alzheimer's are related to beta amyloid metabolism. Familial and sporadic Alzheimer's look identical clinically and neuropathologically. And if you take the duck test, if it looks like a duck, walks like a duck, and quacks like a duck, it is a duck, then beta amyloid must be causing both early onset familial and late onset sporadic Alzheimer's. So in the Baptist versus Taoist competition, the Baptists look to have won. So where has this gotten us? So what became clear when, with those discoveries was that we need to remove beta amyloid. If we can get rid of the plaques, then we should be able to help Alzheimer's. If we block gamma secretase or beta secretase, we can stop production of beta amyloid from APP. And there have been various different drugs tried out. Semagacestat and navagasostat are gamma secretase blockers. Unfortunately, they also cause worsened cognition. Beta secretase inhibitors took a while to develop because it wasn't until 1999 that the enzyme was actually identified and purified. Various different drugs, rubecetat, I can't even pronounce these, but you can see them up there, were tried out, but unfortunately they all cause side effects, worsening cognition and other problems, and have not been successful. The problem with beta and gamma secretase is that as well as cleaving APP, amyloid precursor protein, it also cleaves other transmembrane receptor proteins, including Notch, neurite growth factor, synaptic cell adhesion, and cleaving these can produce a variety of different side effects. These off-target effects may be the explanation for why beta secretase and gamma secretase have problems. Excuse me. Another approach to removing beta amyloid is using immunotherapy. Active immunotherapy, a vaccine to beta amyloid, was developed in 1792. Unfortunately, it was tried in 1999 but abandoned because it caused encephalitis, brain inflammation in some patients. It didn't work clinically, but there was an antibody response. Next to be tried was passive immunotherapy, anti-amyloid monoclonal antibodies. These monoclonal antibodies are artificially made, designer antibodies that attach to specific sites on aggregated misfolded beta amyloid. Monoclonal antibodies are formed by injecting a rat or a rabbit with beta amyloid, isolating the antibodies that are made, picking out the B cell that makes the particular antibody that you want, combining the B cell with myeloma cells, creating a hybridoma that can then secrete and make however many of the monoclonal antibodies as you want. Monoclonal antibodies will bind to the specific site on beta amyloid and then be removed by the immune system. The history of first-generation anti-amyloid mAbs, monoclonal antibodies, was not good. Bapineuzumab was the first to be tried, it failed. Solanezumab was tried in 3 different RCTs and also failed. Gantenerumab was the next one that was tried, 2 phase 3 RCTs, Scarlet Road and Marguerite Road. Both of these were ended early because of interim analyses that looked at, these are early looks into an RCT to figure out whether or not there's any chance that the test drug will actually beat placebo in the RCT, and if it isn't, then the study is ended early. Gantenerumab did reduce amyloid, but it was tried in further more RCTs in Graduate 1 and 2 in 2018, but again failed there. Finally, crenezumab was tried, but also failed to help Alzheimer's. One thing that the monoclonal antibodies did show, was a side effect that's really plagued all of anti-amyloid treatment, and this is ARIA. ARIA-related is amyloid-related imaging abnormalities. ARIA are small regions of edema, swelling, microhemorrhages, bleeding, or siderosis, iron deposits that are seen during anti-amyloid monoclonal antibody treatment on brain magnetic resonance imaging. It turns out that there are amyloid plaques formed in blood vessels as well, leading to congophilic amyloidogenic angiophilia, which is actually a vascular dementia. And what happens with the monoclonal antibodies is that they, first of all, late onset Alzheimer's does have a genetic component, but it's ApoE. It's the epsilon 4 allele of the ApoE gene greatly increases risk of Alzheimer's in late onset Alzheimer's, but doesn't do anything in early onset in familial Alzheimer's. Late onset Alzheimer's doesn't have any indication of problems with APP or presenilin 1 or presenilin 2. And most damning, it turns out that about half of people at their 80s and 90s will have amyloid plaques in their brains but not have dementia. How can you have a cause of dementia and have amyloid plaques in your brain but not be demented? The question came up really was, well, you know the old story about the drunk who's lost his keys and is searching under the street lamp, and he's searching there because, well, the light is better there, but I know I lost it over in the dark there. And the question is that what we were doing with all these anti-amyloid drugs, that we were really looking under the wrong street lamp? One of the big problems with the studies was with the diagnosis of Alzheimer's. Until 2004, the only way to diagnose Alzheimer's was at autopsy. And this was the diagnostic criteria from 1984 to 2011, the NIN-CDS ADRDA criteria, which were used for all of the first-generation anti-amyloid treatments. And the only definitive diagnosis of Alzheimer's required autopsy or biopsy evidence of beta amyloid or tau in the brain. It turns out that about up to a third of patients in the first-generation anti-amyloid monoclonal antibody trials didn't actually have brain amyloid. So they were being treated for brain amyloid, but they didn't have it. There was no way that the anti-amyloid mAb could have worked. What revolutionized things was neuroimaging and the development of PET radiotracers that could actually label and detect the presence of beta amyloid in the brain. So in 2004, C11 Pittsburgh compound B was developed. PIB binds specifically to beta amyloid, and it has a radioactive tag, C11, that can be detected by positron emission tomography. I won't explain how PET works, but suffice it to say that it can produce a picture in the brain of whether or not there's any PIB binding and whether or not there's presence of beta amyloid in the brain. In 2011, various F18, fluorine-18 agents were developed. These are important in being able to use these in clinical trials for a variety of reasons. And in 2011, the Alzheimer's Association and NIH promulgated new research criteria that included biomarkers as part of the diagnosis of Alzheimer's. What happened next was aducanumab, which was a real problem. Amyloid PET was essential to the second-generation anti-amyloid mAb development. They could prove that patients being studied had Alzheimer's to begin with, and they could prove that the beta amyloid had been removed. So aducanumab began in 2018 with PRIME, which is a phase 2, 2012 with PRIME, which is a phase 2 pilot study, and then 2 phase 3 pivotal studies started in 2015. In 2018, The PRIME pilot study read out its results, and it was really, really exciting. It looked like it did definitely remove amyloid. It caused a lot of ARIA, but it looked like progression of disease was slowing in people that had had their amyloid removed. Unfortunately, in 2019, Biogen, the sponsor, canceled the aducanumab trials. They had done an interim analysis on eMERGE and ENGAGE and found that, and the interim analysis said that there was no way that those two pivotal trials were going to come out positive. So Biogen said, well, let's pack up our bags. We already spent, $10 million or $100 million. Let's move on to something else. Unfortunately, or maybe fortunately, when results that were obtained after the interim analysis was started were added, eMERGE turned out to be positive and copied and matched PRIME, showing a beneficial effect of aducanumab. Unfortunately, ENGAGE, the other phase 3 clinical trial, wasn't positive. So Biogen decided to go back to the FDA, and they reopened their new drug application. The FDA asked its advisory committee to take a look at the aducanumab data, and the advisory committee voted 10-0 against the efficacy of aducanumab. But FDA, not recognizing that, well, maybe there's a problem here, decided to give accelerated approval to aducanumab. This caused a huge controversy. The problem is that the efficacy data really weren't consistent. eMERGE worked, ENGAGE didn't, PRIME worked, which is the truth. At the same time, this was the first disease-modifying therapy that might work in a disease for which there was no treatment. At the same time, Biogen was going to make a ton bundle on this drug. They were charging $56,000 a year for the drug. FDA really, the accelerated approval is usually used for cancer drugs, and it's used when there's an early indication that there's a biomarker change that would indicate that the drug should work. FDA thought that removing beta amyloid was an indication that the drug would work, but in fact, a lot of people weren't so sure. And what finally happened was that CMS in 2022 decided that Medicare wasn't going to cover any anti-amyloid drugs that only had accelerated approval, in other words, aducanumab. This is the first time CMS didn't accept FDA judgment on Medicare drugs, but you can imagine it killed aducanumab. The next drug was a bit different. This was Leqembi, Clarity, which was originated by Eisai. Clarity finished in 2022, and the data were consistent. Progression slowed by 27% over 18 months. Amyloid plaques were removed, about 60 centiloids, a large amount. You can see that on the right figure, and you can see the difference in response on the left figure. A 27% improvement means that people on lecanemab were about 5 months behind people on placebo in terms of progression. So that people that had been treated with placebo were 5 months worse, 5 months closer to the end, 5 months closer to death than people that had been treated with aducanumab. This was statistically significant, of course. Unfortunately, ARIA were very common on the drug, 21% of patients, and much higher rates in people that had ApoE4. After Clarity ended, there were actually 3 deaths due to brain hemorrhage that were reported in people that were taking lecanemab. FDA gave full approval to lecanemab in July 2023 with a black box warning of hemorrhage due to anti-amyloid removal. Medicare did decide that it was going to cover lecanemab, and that is the first disease-modifying therapy that's approved and that is available, and that actually does have an effect on Alzheimer's. Another drug, donanemab, was approved in 2024. Progression slowed by 29% over 18 months. Amyloid plaques were completely removed. ARIA was very common, 37%. There were, again, 3 deaths due to brain hemorrhage in patients. And FDA gave full approval in July 2024, so this is less than a year ago. Medicare is going to provide coverage, and both donanemab and lecanemab are being actively used now in academic centers to treat people with early onset, early Alzheimer's, people with mild to moderate, with mild Alzheimer's and mild cognitive impairment. So what do we know about these anti-amyloid monoclonal antibodies? First of all, the fact that they work is really proof that beta amyloid is involved in both familial and late-life Alzheimer's. There's really no question in familial Alzheimer's because the mutations were affecting beta amyloid. If the mutations were affecting beta amyloid and causing Alzheimer's, then that was the cause of Alzheimer's in late life. This, though, the anti-amyloid monoclonal antibodies were necessary to show that beta amyloid was also important in late life Alzheimer's. Late life and familial Alzheimer's have the same exact plaques and tangles, the same presentation except for the timing, and it turns out they really do have the same disease process. Anti-amyloid monoclonals remove beta amyloid, slow progression, and modify disease. Most importantly, they aren't cures, but they buy time. The problem is with ARIA, which is potentially fatal, and they're incredibly expensive, $26,000 a year or $32,000 a year for 1 year of therapy. What is really important, though, is that they showed that we were looking under the right street lamp, that we were looking for the right thing. But the results were really, it was glass half empty or half full. And it can be interpreted either way. It's very reasonable to question, what is really how good are these? Do you really want to be using this drug that may kill you and that is only going to buy you a few months? But at the same time, do you want to use the only drug that might actually cause a change in your illness and might slow down your illness? And, the Baptists and Taoists really have pretty much resolved their differences, and now it's clear that both beta amyloid and tau are important in Alzheimer's. I'll be talking this more lately, but more competition, these insignias are for Real Madrid and Barcelona, so it's not the Sox and the Yankees, but it's the same idea. Scientists, when they get an idea, when they believe in something, they believe in something. Otherwise, they wouldn't work on it. Now, there's preclinical Alzheimer's, and this is really important. It turns out that amyloid plaques appear 20 years before dementia begins. This was only possible to be seen with the development of amyloid PET, because amyloid PET could be used when people were alive. You couldn't possibly see this effect in a postmortem study, but what amyloid PET showed was that beta amyloid plaques start accumulating, but dementia doesn't happen for another 20 years. When it does happen, it turns out, now the actual starting time can vary greatly, but it turns out that the rate of progression once accumulation does start is very similar across people, and you can actually develop an amyloid clock that can point to when your amyloid first started accumulating. So there are 2 important things to be learned from this. First, it's not surprising that many people with beta amyloid in their brains don't have dementia, and that they die and they're fine, and you see beta amyloid because they died before they got dementia. The other thing that it shows is that beta amyloid cannot be the primary driver of cognitive impairment. There's this 20-year break Between when beta amyloid appears, it accumulates for 20 years, and only then does dementia actually present. So what is the culprit? The culprit are tau tangles. That's the other landmark feature that Alzheimer's described. Remember, the normal function of tau is to stabilize microtubules, but tau can misfold and aggregate into fibrils and tangles. Alzheimer's is a tauopathy, but unlike other tauopathies, it includes both amyloid and tau. The other tauopathies are purely due to tau, 4R tau, 3R tau, Pick's disease, progressive supranuclear palsy, frontotemporal dementia. So now I'm going to talk about the brain. This is hopefully not too confusing. This is a midline view of the right hemisphere of the brain. There are 4 lobes, frontal, occipital, parietal, and temporal. These 4 lobes make up the neocortex, which are the most developed part of the human brain. The hippocampus is also important. The hippocampus is deep within the temporal cortex, temporal lobe, and is involved in Alzheimer's. In 1990, 2 German neuropathologists, Heiko and Eva Brock, examined more than 2,000 brains from all ages and described normal tau accumulation in aging and in Alzheimer's. And what they found was that there's stereotyped progress. Tau begins in stages Brock 1 and 2, transentorhinal medial temporal cortex. You can see that at the very tip of the temporal cortex in the amygdala and the hippocampus. It starts spreading into stage 3 and 4, where it starts moving into the rest of the temporal lobe and then spreads into the rest of the hippocampus, which actually goes around in the perihippocampal gyrus up to the fornix. And it's during Brock stages 3 and 4 that dementia starts being diagnosed. Finally, in Brock stages 5 and 6, the tau tangles spread to the rest of the neocortex, the visual in frontal, parietal, and occipital lobe, and dementia occurs. The specific symptoms will depend on where the tau tangles appear. If they're in frontal cortex, you'll get behavioral and dysexecutive problems. If it's in occipital cortex, you'll get more speech problems. In occipital cortex, you'll see visual spatial problems. Or parietal cortex, you get speech problems. Everyone eventually develops tangles. As we get older, everyone is at Brock 1 or 2 or a later phase. This is virtually everybody. So Baptist and Taoists had made peace. Both beta amyloid and tau are needed for Alzheimer's. This is the neuropathological criteria that are required for Alzheimer's diagnosis. Now, PET scans are used instead of postmortem, although postmortems can still be used. And what this figure shows is that you have to have a lot of tau and a lot of amyloid to be diagnosed as having Alzheimer's neuropathology. And having Alzheimer's neuropathology doesn't actually prove that you have Alzheimer's disease, Alzheimer's dementia. This is an autopsy study from the United Kingdom that was done in 2009 that really tells us a lot about Alzheimer's. There are 2 brain regions, hippocampus and neocortex, top and bottom, and it looks at 3 different biomarkers, plaques, tangles, and atrophy. And it compares people with dementia in blue to people without dementia. And what you can see is that, and it follows people as they age. So the horizontal axis is time, is age. And what you can see is that everyone's brains atrophy with time. But people with dementia have more atrophy in both the hippocampus and the neocortex. Both dementia and healthy controls have both plaques and tangles, but there are many more in dementia than in people with than people that don't have dementia. Hippocampal tangles are present in everyone that develops dementia. And in fact, they become pretty common in healthy people at a late age. That would be like Braak stages 1 and 2, maybe going into 3 and 4. But what you can see is in the neocortex, tau tangles aren't very common in people that don't have dementia. They're only common in people that have dementia. You'll see that, the importance of this shortly. So tau PET was developed in 2013. 3 tau tracers were developed and became available and were used. It's possible to do Braak staging with tau PET. Using tau PET and amyloid PET, we've developed a new model for Alzheimer's disease, the ATN model. We can diagnose Alzheimer's while people are alive, and we can see both beta amyloid and tau in brain. What happens first is amyloid comes first, A, in the neocortex and then gradually spreading. There's no dementia, though, until tau starts being seen outside of medial temporal cortex and being spread. And then finally, as tau continues to spread and amyloid continues to increase, the brain starts to atrophy and you get neurodegeneration. And this is the model that we're currently using for Alzheimer's disease. It seems to have frozen. Okay. So how are amyloid and tau connected? I mean, they're both involved. It's kind of complicated. Amyloid comes first, and there's much more beta amyloid than tau. In the study on the left from ADNI, you can see that there is some tau in impaired people that don't have amyloid, but much more tau in people that are impaired and have amyloid present. And the study on the right from the Harvard Aging Brain Study shows that baseline medial temporal lobe tau and amount of amyloid binding predicts subsequent change in inferior temporal tau levels. So what predicts dementia and predicts spread of tau is the presence of amyloid and the presence of tau. It builds on itself. This study is a sort of a meta-analysis involving 6 different Alzheimer's cohorts, and it shows very importantly that what's really important is the spread of tau. So what they looked at was all amyloid, tau, and neurodegeneration, ATN staging. And what they see is that in people with negative amyloid, negative tau, there's no dementia. People with positive amyloid, positive tau, there's some mild cognitive decline, but no dementia. In people that have medial temporal tau and amyloid, you start to see dementia. And if you have people with neocortical amyloid and tau, that's when you really see dementia. So it really is the spread of tau from the medial temporal lobe to the neocortex that really matters the most. Alzheimer's has 2 phases. There's a lengthy pre-symptomatic phase while amyloid plaques accumulate. Braak 1 and 2 and medial temporal tau appear. But then there's a symptomatic phase when tau spreads from medial temporal to temporal and the rest of the hippocampus and then to the rest of cortex. It's not quite that simple, since amyloid and tau accumulation are really separate processes. Plaques continue to accumulate while tau accumulates, but Braak 1 and 2 are tau, but they're pre-symptomatic. It appears that plaques somehow drive tangles. Do plaques and tangles independently cause damage, or is there some process that drives both? It also raises the question of when should Alzheimer's treatment begin? Treatment trials have all been done during the symptomatic phase. What would happen if treatment started during the pre-symptomatic phase? Should someone without dementia be diagnosed with Alzheimer's? Could removal of amyloid plaques, it's clear that having amyloid present is a risk factor. You're on the path to developing dementia, but it may be years and years away. So does it make sense to diagnose someone then with dementia? Only if removal of amyloid plaques is protective. And that actually is being studied, or is being studied. So this is not the study that I'm going to be talking about, but it's an earlier study. This was a study of solanezumab, which you might remember was one of the first generation anti-amyloid monoclonal antibodies, but this was looking at preclinical Alzheimer's. So these are people that are healthy controls that have amyloid in their brains. They don't have any dementia. And what you see in this study, this is the A4 study, is that over a 4-year period, both groups decline. There's no difference between placebo and solanezumab, just like solanezumab didn't work in people that had early Alzheimer's. But what it does show is that in 4 years, you can pick out decline so that you should be able to see something that is different, something that's different from placebo. You should also be able to see changes in tau. And donanemab slows tau. Both donanemab and lecanemab have been shown to slow tau accumulation. So in theory, these drugs should be slowing tau accumulation. Maybe if you give them during the asymptomatic phase, they'll prevent tau accumulation and prevent dementia. And there are 2 studies that are actually looking at this. AHEAD is the first study using lecanemab, Leqembi, and is studying, it's looking at 2 groups of A4-5 and A3, people with higher amyloid levels and lower amyloid levels. The dose of amyloid of Leqembi varies depending on whether it's higher or lower. They're followed for 4 years, and they're going to see whether or not people that get to treatment develop dementia, develop cognitive impairment, or if they do better than people without dementia, without amyloid in their brains. Trailblazer-Waltz 3 is looking at donanemab, doing the same sort of study. Both studies, so people have intact cognition, but they have amyloid present in their brains, and the studies will test whether or not removing the amyloid will actually prevent subsequent onset of dementia. It improves progression. It slows down progression. But what's it going to do before progression has started? And that's really the interesting question here. There's another study, this is very preliminary, but this is from the Diane study. I told you we have a cohort of patients with dominantly inherited Alzheimer's disease, and this was a treatment study in people that didn't yet have dementia, but had the gene, had the genetic mutation that was going to cause dementia. One of the interesting factors with familial Alzheimer's is that the time of onset tends to run in families. So if your father had his dementia start around age 50, there's a very good chance that your dementia is going to start around age 50 as well. In the Diane TU study, they compared solanezumab to gantenerumab. Gantenerumab does remove amyloid, and what they showed in a preliminary study, this is very preliminary, is that in these people that would normally for sure develop Alzheimer's, they really have a death sentence, that treatment with gantenerumab actually prolonged their time disease-free. They're going to continue these patients, and we're going to continue to see what happens. So what's the take-home? What does all this mean? So Alzheimer's involves both beta amyloid and tau, and it can be diagnosed in living people. There's a pre-symptomatic phase while beta amyloid plaques accumulate, followed by a symptomatic phase when tau tangles spread. Anti-amyloid monoclonals can remove amyloid plaques, and they buy time for patients during the symptomatic phase, but there's a risk of death with ARIA. Familial and late onset Alzheimer's share a common pathology. The amyloid model was correct but incomplete. And can removing amyloid plaques prevent onset of dementia? And maybe most important, we don't know how AHEAD and Trailblazer all three are going to turn out, but we now have a complete framework for understanding Alzheimer's. We know that both amyloid and tau are involved, and this provides a framework for developing more studies and understanding the actual details of what's happening. Things are going to look very different if we can prevent dementia in people at risk. We're now able to diagnose Alzheimer's using positron emission tomography or using LPs to gather CSF. Both of those are pretty invasive, and I don't imagine that's something that most people will want to do, especially if they don't have symptoms. However, we've recently discovered that there is a blood biomarker, phospho-tau, phosphorylated at amino acid 217, actually looks like it can substitute for amyloid PET or CSF. And in fact, p-tau 217 is being used in both AHEAD and in Trailblazer ALTS 3. Anyway, the hope is that maybe in 2030, the trials end in 2028. In 2030, maybe we'll be able to go and get our blood drawn. I'm going to get my blood drawn to check my p-tau 217 levels and see if I have amyloid in my brain. Hopefully I won't be demented at that point, but if I do, I'm going to think seriously about getting that amyloid removed. There's so much more that needs to be done. It's kind of like cancer and being able to diagnose cancer early. Diagnosing cancer early has been really, really important, and it may be really important in Alzheimer's as well. Anyway, that is my talk. I'm sorry I was kind of incoherent, and I've taken up my time, but any questions? Thank you. Yeah.
And ask them to wait for the mic.
I think you're going to need to yell.
Yeah. No. Please raise your hand if you want to ask a question. Okay.
Yeah.
Bring the mic down here, please.
The gentleman in the blue shirt who's going to yell very loudly. What's your question? Oh, here comes the microphone.
Rusty asked a question.
You need to turn it on.
Question I have for all the clinical studies is how have they addressed the blood-brain barrier problem that prevents large proteins like monoclonal antibodies from getting into the brain?
You know, I need to get hearing aids. You all know about that. But so
Okay, I'll go louder. Is this better?
Yeah, it's the echo that gets me.
I'm just wanting to know how these clinical studies address the problem of the blood-brain barrier preventing these monoclonal antibodies from getting into the brain.
That is super important because these monoclonal antibodies don't get into the brain very easily. You have to give really big doses of them. That may be a big part of why you get ARIA, because you get a whole lot more monoclonal antibody inside the blood vessel, binding to the beta amyloid in the blood vessel before getting into the brain. But there's a brand new drug, Roche has, that uses gantenerumab that's tagged with transferrin, and there's a transferrin transporter in the blood-brain barrier that can take things that are bound to transfer it into the brain. And pilot studies show that this brain shuttle technique can clear amyloid very effectively with much, much lower doses. Whether or not it's going to continue to work, whether or not there's going to be other problems, you know, who knows? But that's there.
Yeah. You told us quite a bit about, I'm over here on your left side. Hello. You told us about how beta amyloid was formed, but less about tau. And it looked like from your histology that it's within the neurons that this abnormal protein that is formed,
So
the
you're-
abnormal tangles. Do we know how that happens and why?
So it is important that amyloid actually is extracellular, so it's not in cells. Oh, and one important thing that I forgot to mention is there is no dementia that's due to amyloid alone. There are lots of dementias that are due to tau, and Alzheimer's is a tauopathy, but it's a tauopathy caused by amyloid. Tau, on the other hand, is within the neuron. Why does it accumulate? How does that happen? I wish I knew. And we will learn. We will find out. Those studies are being done. There were a huge number of studies done in the 1990s when, we first found out about the amyloid hypothesis and, tau, and looking at how amyloid drives tau, and it has something to do with phosphorylation of tau. Tau is controlled by phosphorylation, so it turns out that when you fall asleep, your tau gets phosphorylated. But when you wake up, the phosphates are taken off. When people develop dementia, tau becomes irreversibly hyperphosphorylated, and that's when it comes together, forms these tangles, and causes dementia. But there's still a lot more going on in there.
Now that you have these nice methods to look at levels of tau and amyloid, is anyone looking at how lifestyles might change that? Exercise, drinking, whatever.
Talk to the mic.
Oh, I'm sorry. Lifestyle is important. I mean, it's not important for amyloid and tau. It's important for life. And it will only add to, or let me rephrase that. If the anti-amyloid monoclonal, if removing amyloid works, that's only going to add to a good lifestyle, a healthy lifestyle. Keeping busy physically, keeping busy mentally, taking care of your family, being socially active, coming to things like this, listening to me talk. That's all good for you.
I know somebody who is 60, and her mother had Alzheimer's, I think, for a number of years. And she seems very alert and active. Is there anything that her age that she can do to maybe, she's very active, is that about it?
I am sure she's doing all the things that she needs to do and can do, and that you're helping her do them. As people get older, actually, amyloid and tau get less important, so that people in their 80s and 90s have a whole range of neuropathologies. So it's really not just amyloid and tau. I mean, they may still be called Alzheimer's, and they still may be treated, and it may still be useful, but it's really much more complicated.
How long will the experiments, these magnificent experiments, will this kind of funding be kept up by the federal government?
I haven't been fired yet. Sometimes I think about quitting, but that's different. So the studies of the AHEAD and Trailblazer All 3 are actually mostly being paid for by the drug companies. They're the ones that are really going to benefit. That actually is kind of the problem, because does anybody here trust the drug companies? And it may turn out that they're actually doing it's. So you've seen these ads in TV from Lilly for, is it that accumulation of amyloid is bad? They're setting us up for donanemab, for getting amyloid removed, And the thing is, they're telling the truth for a change.
One more question. Okay, well, thank you very much.
Thank you.